If you want to score a 520, you have to know this topic.
So whether you're preparing for the MCAT or getting ready to take your first bio course, one of the major things we have to understand are the differences between prokaryotes and eukaryotes. Prokaryotes can be further divided into bacteria and archaea, and then eukaryotes include animals, plants, fungi, protozoa, right, to name a few. And essentially, these are the two major domains of life, right? Now, there's a lot of differences between these, but the fundamental difference is that prokaryotes do not have membrane-bound organelles. Essentially, they are just a cell membrane containing cytoplasm with a lot of different processes happening inside. Whereas eukaryotes, all of these different organisms, they are going to have membrane-bound organelles. So they have the cell membrane, and then enclosed within that membrane is the sea of fluid and these organelles within that fluid. Those are the membrane-bound organelles, and the fluid is the cytoplasm. And the organelles themselves, they have their own membranes, and what this results in is compartmentalization, where there can be separate processes that are occurring isolated within those organelles, isolated from the outside environment, okay? And that has interesting implications. Okay, now we're gonna discuss in more detail the high yield differences between these two. Okay, so now let's compare and contrast these three. So first of all, do they have organelles? Well, bacteria and archaea, by definition, do not have organelles as they are prokaryotes, but eukaryotes do. Okay. Now, organelles are membrane bound. So while bacteria and archaea have a cell membrane, they do not have any membranes within the cell, right? Which means that all the processes that are happening inside these cells, like all the metabolic processes, these different pathways that we learn about, are all happening in the same environment. They are not compartmentalized. However, in eukaryotes, which do have membrane-bound organelles, these processes can be compartmentalized, which is very important for their regulation. Now, another difference is the type of chromosome. So first of all, bacteria and archaea usually have only one single chromosome, and those chromosomes are circular. Okay, so these are circular chromosomes, and this applies to the archaea. Eukaryotes, however, have linear chromosomes, and eukaryotic cells, of course, like our cells, can have numerous chromosomes instead of just one. Now, there are examples of archaea having multiple chromosomes, but for generalization purposes, we're going to say that prokaryotes have one circular chromosome, whereas eukaryotes can have multiple linear chromosomes. Like, for example, our cells have 46 chromosomes, 2 pairs of 23. Okay, one from the father, one from the mother. Now, one of the consequences of this is that these circular chromosomes, when the cells divide, do not shorten because they're just a circle. Whereas eukaryotes, because they have the linear chromosome, the ends of these chromosomes can be degraded, and some of that information is lost with every cell division. And so the mechanism that we have in our cells that prevents the loss of these chromosomes, or at least decelerates, makes slower the loss of these chromosomes, is of course the enzyme telomerase, which helps to sort of save some of that chromosomal length to make the aging process slower, okay? Because aging is actually related to shortening of the chromosomes. Now, another difference is the presence of histone proteins. Remember, histone proteins are those positively charged proteins that bind to DNA. Now, the thing is, bacteria do not have histones, so the bacterial DNA does not bind to histone proteins. However, some archaea do have some histones. So we're gonna say that archaea does, and this is actually one similarity between archaea and eukaryotes, because eukaryotes, of course, also have histones. And remember that the histones will package together with the DNA. The histones are positively charged, the DNA is negatively charged. They will bind together, and that will form a structure called chromatin, which will fold even more to form the chromosome. Now, cell wall, the presence of a cell wall. Bacteria do have cell walls. Now, of course, depending on the type of bacteria, gram-positive, gram-negative, the cell wall is kind of different, but they do have cell walls. Whereas archaea actually do not have cell walls. And then eukaryotes, well, eukaryotes sometimes have cell walls, right? For example, plant cells do have cell walls, but archaea do not, bacteria do, okay? Now, another interesting difference is the type of membrane lipid. Remember that the cell membranes, and then also in eukaryotes, the organelle membranes, they are composed of lipids, and then also proteins and carbohydrates and things like that, right? Now, the typical membrane lipid in bacteria is basically like your fatty acid. Now, a fatty acid, for those of you that don't know, is actually an ester-linked. It has an ester linkage, okay, between the glycerol head group and then the fatty acid. So I'm going to say that these are esters, okay? And in eukaryotes, it's the same thing. They're also esters, okay? Now, archaea, on the other hand, and this is a difference that makes archaea unique, their membrane lipids are actually ethers, which look like this, okay, versus an ester where usually it looks something like this, okay? And so, again, I'm not going to get into the specifics of the type of ethers or esters, but remember that in bacteria and eukaryotes, it's an ester, like your typical fatty acid, whereas in archaea, it's actually an ether. Now, bacteria and archaea as prokaryotes, their ribosome, which remember is that machinery, that RNA plus protein complex that actually reads the messenger RNA and synthesizes protein from that, right? It's basically that assembly line for protein production. Now, the ribosomes, there's a small subunit and a large subunit, right? In bacteria and archaea, right, they have a small subunit, which is 30S, and then a large subunit, which is 50S, and that forms a 70S ribosome complex. And this is true for both bacteria and archaea, 70S, okay? Now remember, don't make the mistake of getting tricked saying that 30S plus 50S is 80S, right? This is a kind of a weird like unit that they're using, sedimentation units, and you do not add up the numbers. You just have to memorize that the small subunit is 30S, the large subunit is 50S, and the complex, when they come together, is 70S. And that's true for archaea and bacteria, so the prokaryotes. Eukaryotes, however, have a small subunit that's 40S, and then a large subunit that's 60S, and again, kind of annoying, but this makes, it actually doesn't make 100S, right? That's my mistake. This actually makes an 80s, right? So I almost fell for it myself, but it's an 80s, okay? So just remember that it's 10 more than the bacterial one, okay? But it's 40 plus 60 for 80 and 30 plus 50 for 70, okay? Now, another difference is that bacteria, remember, they do not have introns, okay? Now, introns are basically regions of DNA that do not code for protein, okay? And bacteria just don't have introns at all. And this has the implication that when, when you actually transcribe the DNA, you produce mRNA directly. Okay, so and that mRNA can be immediately translated in the cytoplasm because all of this is happening in the cytoplasm for bacteria and actually Archaea. Archaea, however, do have some introns. Okay, so another similarity between them and eukaryotes. And eukaryotes, of course, have many introns. Okay, so they have a lot of introns. And remember, those introns have to be spliced out, and then the exons have to be spliced together to form that continuous mRNA. Okay, and of course that allows for the process of alternative splicing, where you can get different combinations of exons to produce slightly different enzymes or proteins called isoenzymes. Now, the type of cellular reproduction, how do these cells reproduce themselves? So bacteria and archaea are going to be reproducing through the process of binary fission, which is asexual reproduction. Whereas eukaryotes, they can reproduce asexually through mitosis, okay? And then also they can reproduce through meiosis, which is sexual reproduction, okay, meiosis, which has 2 steps. And so that allows for genetic variability that can be passed on through vertical transmission, okay? Now, those are sort of the high yield differences. A couple of other caveats that I want to note, right? So first of all, when it comes to the chromosomes, remember that while bacteria and archaea generally have just one circular chromosome, they can also have commonly extra chromosomal DNA, especially bacteria. They can have plasmids, and those plasmids can be transferred between the bacteria through horizontal gene transfer mechanisms such as conjugation, transformation, transduction, okay? Eukaryotes, however, typically do not have plasmids. However, you can give a eukaryotic cell a plasmid, and that's usually done in like a laboratory to try to express certain genes, okay? That's one difference. Now, another important thing to remember, and specifically for archaea, and again, this isn't a very high yield topic, archaea, but something just to keep in mind, these are kind of interesting organisms. They tend to be what are called extremophiles. Extremophiles are organisms that are capable of surviving in very extreme conditions, such as extreme heat, for instance, or very salty environments or very acidic environments, things like that. So archaea tend to be extremophiles. And actually, for those of you that are aware of the so-called polymerase chain reaction, which is commonly used in laboratories to multiply DNA, that process uses a special type of DNA polymerase, which is called the Taq polymerase, TAQ. And the Taq polymerase is actually isolated from archaea. And the reason is because that polymerase needs to be very heat stable, because one of the steps of PCR is to increase the temperature substantially, but you don't want to denature and make dysfunctional the DNA polymerase, so you need a heat stable one, so that's actually isolated from a thermophilic archaea, the Taq polymerase.